Well, good morning, ladies and gentlemen, dear colleagues. I would like to thank the organizers for inviting me to the BSC annual meeting. I'm very happy to present some details about pyrrolizidine alkaloids in Europe, pyrrolizidine alkaloids, or shortly PA. In particular, the industry's initiatives as well as the regulatory background. At first, some general remarks about PA and why all the activities related to patient safety and consumer protection are so important. PA are naturally occurring substances in plants in specific families, for instance, the Asteraceae or the Boraginaceae. They have a basic structure of a nicene body, and they form ester alkaloids that occur as monoesters or open or cyclic diesters. The problem is that long-term exposure to PA primarily damages the liver, the lungs, and the blood vessels. PA are also genotoxic and carcinogenic. The cyclic diesters of the 1,2 unsaturated PA are considered to be the most toxic, followed by other structures. And chronic exposure to low levels might lead to accumulation of protein adducts and liver damage. But that's only as a brief introduction why the discussion is so important. I think my colleague Catherine Mahony will talk later on in more detail about the toxicology of these substances. Our current regulatory discussion started in 2013, about 10 years ago, about or when the German Federal Institute, the BFR, Federal Institute for Risk Assessment, this is a health authority in the food sector, performed a study where they found PA in herbal teas produced from plants which normally do not at all contain PA. And then the discussion arose whether this could be a problem of contamination. And as a source of contamination, co-harvesting with toxic weeds growing in the same fields like medicinal plants were suspected or identified, with severe consequences for herbal medicinal products. So from the viewpoint of the German herbal industry, PA contamination in medicinal plants and their products was considered a very serious problem and also a challenge for agriculture and horticulture in general. Interdisciplinary solutions were needed along the entire process chain. That's why from the beginning already, the cultivators and their associations were involved in the entire discussion. However, weeds, including, for instance, the Senecio, the Jacobea, the Crotalaria, do not present a new phenomenon. I assume they have always grown somewhere in the fields. However, PA are nowadays detectable already at trace levels because the analytical methods are becoming more and more sensitive. Rather soon, the herbal industry, together with the farmers who cultivate medicinal plants, undertook a lot of measures in order to minimize the content of PA in herbal medicinal products. This comprised causal research and testing, collection of analytical data, participation in research projects, and establishing a code of practice. On the authorities side, the problem was addressed as well, and the health authorities, particularly the Herbal Medicinal Products Committee of the European Medicines Agency, the EMA, developed a public statement of herbal medicinal products containing PA. This was originally made for PA containing plants, but due to the newly arising discussion on contaminants, also the problem of contamination was taken into account and became part of this statement of the HMPC. There was a long discussion about the maximum daily intake of PA. In the beginning, they discussed 0.035 microgram PA as a maximum daily intake, then coming to the 10th-fold amount, and finally to the 1.0 microgram per day. The extent of testing addressed to the manufacturers was defined. This was identical to measures undertaken by the German bee farm in 2016. A risk assessment had to be undertaken for the definition of the extent of testing. That means either skip testing or much narrower skip testing in the first two categories, or even a routine testing if there were no data available or if the risk was considered very high, and that means routine testing of every batch. Now, I would like to talk a little bit about this code of practice, which has been developed by herb growers and the herbal industry. It was published in a journal, in the Journal of Applied Research of Medicinal and Aromatic Plants. And it was designed to be a guidance document for the herbal industry, for the companies who then could develop individual or product-specific actions in order to reduce the risk of PA contamination as far as possible, always in cooperation with the herb growers. The background was that, of course, a complete avoidance of PA contamination was impossible according to the current technical state of the art. Thus, preventing measures in agriculture, monitoring and testing of batches was regarded essential. The principle of this code of practice is that a risk analysis should be performed for each production step along the entire process chain, and proposals for corresponding actions were made. And in the following, I would like to show you some examples. You can see the first step of the entire process chain, the cultivation or even the cultivation planning and the seeding. And for each of these steps, the risks were described, what can happen, the probability of the risk, several options to exert influence to reduce this risk, and finally, the feasibility, the time horizon, and the responsibilities. Some examples, for instance, during a cultivation or cultivation planning, it can be an option to select fields without corresponding weed populations, to observe a crop rotation, to pay attention to cleanliness of the equipment after changing fields, and so on. Another example is, for instance, the weed infestation in the field. What can be done here? The cultivation method and the species-specific actions in the weed management should be taken into account, and/or selective crop protection measures can be undertaken, or the manual or mechanical weed control can be intensified. In one of the further steps, the incoming goods inspection of the crude drugs in the pharmaceutical company. Here it is important that a risk-based selection of crude drugs which are subject to intensive testing for PA. Those drugs which have a high risk of being contaminated should be done. And the same applies then to the release of the active substance before it goes into production of the final medicinal product. Coming to regulatory actions of the health authorities, in July 2021, the already mentioned HMPC of the European Medicines Agency finalized and published their public statement on the use of herbal medicinal products containing toxic unsaturated PA. And one of the most important issues in this document is that they fixed a limit for the daily intake of PA through medicinal products of 1.0 microgram for adults. For children and adolescents, the intake should be calculated according to body weight. Well, there is one exemption which is mentioned in the statement, that the product group of essential oils is seen to have a very low concern. So essential oils which have a pharmaceutical quality contained in HMPs would not need to provide specifications concerning PA content. All other products have to do so, but of course a risk assessment is possible, but this must be documented, and in particular batch data with PA analysis have to be submitted to the health authorities. Well, with regard to analytical determination of PA, in former years, the Code of Practice already recommended the BFR procedure, which was established in 2014 as state-of-the-art technology, but later on, the European Pharmacopoeia developed a general chapter 2826 on contaminant pyrrolizidine alkaloids, which was published and implemented in January of last year. According to this general monograph, the use of any procedure, chromatography coupled with MS-MS or high resolution MS, is possible if specific validation criteria are met, but the Pharmacopoeia monograph implements almost completely the already existing procedure published by the BFR. And then there is the possibility of a matrix-specific validation, which I would not describe in detail at the moment. Well, with regard to this procedure, the extent of testing comprises 28 substances, which are mentioned here as an overview. And what is rather new now, this monograph containing the analytical method will now be included in the general Pharmacopoeia monograph on pharmaceutical preparations, and will therefore be made compulsory because it's mentioned in this monograph as a test which has to be applied if necessary. For instance, for medicinal plants which are suspected to contain PA. In this context of the analytical testing, the issue of sampling is also very important. The Pharmacopoeia monograph says that a uniform particle size and a homogeneous distribution of PA is important. An inhomogeneity can be caused because PA contamination caused by individual PA-containing plants growing in the fields is a spot contamination. That means before we do the analytics, we do really need a homogeneous distribution in order to receive reliable result. The European Pharmacopoeia monograph does not say anything about sampling, but in practice there are different possibilities. The first one is the European Pharmacopoeia chapter 2A20, or the more extensive sampling is the one according to the European regulation on mycotoxin determination, which also can be used. And in herbal industry, normally a risk-based approach is made, and then one of these procedures is chosen. Moreover, the European Herb Growers Association, EuroPAM, is working on a sampling proposal for high-risk lots of medicinal and aromatic plants with spot contaminations of PA. It's not yet published, but I think it's rather close to finalization and publication. And here they give really valuable advice on how sampling before PA testing should be performed from a cultivator's viewpoint. Well, according to industry's experience, the situation on PA contamination is, of course, observed. This is done by data evaluation and data collection, which has started in 2013. Approximately 50 companies have collected all their data and put them into a joint database comprising herbal drugs and herbal extracts. Regarding the products with the highest market relevance, we have seen a remarkable improvement during the first years, from 2013 to, let me say, 2017-18. And nowadays we have a stable situation with regard to the herbal drugs. You see, if you have a look into the red columns on the first figure, which describes the herbal drugs, the red column is going down a bit and is stable now. This is the these are the batches with which have more than 1.0 microgram, always calculated to the final product. And in terms of the extract, we can see that the situation is much better, that we are somewhere around 0, but with slight oscillations every year. You can also see that the PA contamination cannot be reduced completely. This is simply due to the fact that sometimes there are, of course, weeds growing in the fields where medicinal plants are grown, and they cannot be deleted completely from the fields. However, it's important to say that in any case, the 1.0 limit has to be met. If it's not met, a batch cannot be used for further production, but has to be rejected. We can, however, from this data evaluation, see that in many cases, the daily limit of 1.0 microgram PA related to the final product can be kept. And that is the reason why the HMPC limit seems rather appropriate. We would have faced bigger problems if they would have chosen a limit much lower than 1.0 microgram per day. Well, that was the medicines area. I would like to say some words on the food area. as food products and the ingredients for food products which are of herbal origin also grow in fields. This is also PA plants might also present a problem to food products. I would like to give you some examples. A study of 2018 in 6 European countries showed that in several products, milks, eggs, meat, there were PA findings only in very few cases, but in 60% of herbal food supplements examined and in more than 90% of herbal teas used in the food area, measurable if amounts of PA were found. Then honey is of course a problem because you never know where the bees collect their products, and that is the reason why the EFSA has in 2011, EFSA is the European Food Safety Authority, has identified health risks when big amounts of honey are consumed. And there are some studies on honey from Germany, from the environment where ragwort, Senecio grows. There were significantly higher PA levels than those from areas without ragwort. Well, coming to the food legislation, there is the European regulation on contaminants in food, the former 1881 from 2006. This has now been transferred into the new regulation from 2023. The amendment that has been made in December 2020 concerns maximum PA levels in food products for certain teas, herbal infusions, but also for food supplements. But these are in the same magnitude as the limits for the medicines area. The limits for the food area are given in microgram per kilogram, but if you calculate it using the daily intake, you will arrive somewhere in the same area as the limits in the medicines area. With regard to the analytics, these levels refer to the lower bound sum of 21 alkaloids. This is a little bit different from the pharmacopoeia area, but additional 14 alkaloids can be determined and then included in the total sum as well. Well, one word on the toxicology, and this is already the link to Catherine's presentation that you will hear soon. It has been shown that different PA have significantly different toxicological properties. I think Catherine will talk about that in detail. I would just like to mention that in the past few years, altogether 3 workshops have been performed, 2018, 2020 and 2022, in cooperation with the Technical University of Kaiserslautern, and all these results are published. The last one, I think the September 2022, is already on its way and will be published soon in Planta Medica. Well, as a summary, I would like to say that in order to minimize PA contamination, herb growers and herbal industry have implemented their code of practice. The health authorities ask for assessment and control, sometimes, of course, routine control if it's justified. The EMA HMPC has set a limit of 1.0 microgram PA per daily intake through herbal medicinal products. But nonetheless, we have to say that contamination of plant material with PA continues posing a big challenge for growers as well as for manufacturers. Since 2 years, we have the PHOIA, the European Pharmacopoeia method for a precise determination of contaminant PA. And regarding the data that I've shown, after several years of continuous reduction of the PA content for herbal drugs and extracts, a stable situation has been achieved now over the past 5 years. And with this summary, I would like to thank you for listening. And if you have any questions, we can discuss them after Catherine's presentation or later on. Please do not hesitate to send me an email. Thank you.
Many thanks, Barbara. That was a really excellent talk and helping set the stage on PAs. I do ask we'll hold questions for the end with you and with Catherine. So next, we're going to have another one of our steering team members introduce Catherine. So Amy, if you're on.
Yes, thank you, Connie. It is my pleasure to introduce our next speaker, Dr. Catherine Mahony, who is a colleague and a friend. She is a research fellow in our corporate safety organization at Procter & Gamble, and she has been working for 25 years in ingredient and product safety, which includes, in particular, botanical safety and natural ingredient safety. And she really has pioneered a lot of the approaches, particularly the new approaches to assess risk assessment, including botanical safety for us here at P&G. And she is a founding member of the Botanical Safety Consortium, and she co-chairs the Botanical Safety Consortium Developmental and Reproductive Toxicology Working Group. So with that, I will turn the virtual floor over to Catherine for her presentation this morning. Thank you, Catherine.
Thank you, Amy, for such a kind introduction, and thank you to the BSC for inviting me. I mean, Barbara's given a really nice background to this topic of pyrrolizidine alkaloids, and yes, to make life easy, let's just call them PAs. So it's a field that we've been working on in P&G for the last 5 years. I'll say a little bit more about why, but I think it's a really nice example of how you can use in vitro data for, okay, granted, in our case, relatively simple mixtures of these PAs versus the complexities of botanicals that maybe the BSC are working with. But yeah, so I think it's a nice visualization of what can be done with in vitro data, and in our case, really to aid the risk assessment of this PA issue that Barbara so nicely introduced. So the limits that Barbara highlighted are really based around a potent PA, and yet we begin to see already in 2016 that maybe not all PAs are created equal. We know that PAs have to have this double bond between the 1 and 2 carbon position in order to be toxic. But we also see from this first proposal by Merz and Schrenk that there are some other factors that can drive toxicity differences between PAs. So we have these nasic acids. We can either have diesters or monoesters, and these can either be in an open or a closed configuration, and the carbon at the 7 position can be in either the R or the S configuration. So all of these factors combined really makes for some differing potencies across different classes of PAs. So that's relevant in that yes, we must have as low as reasonably achievable levels of PAs, but we can also start to ask the question, what is toxicologically relevant for the risk assessment? So I wanted to just spend a minute or two talking about what we know to be the driver for toxicity of PAs. So PAs have to be metabolized in order to form reactive intermediates, reactive metabolites, which can then undergo either further metabolism or directly bind to DNA. So metabolism in this case is activating for the toxicity of PAs. So regardless of the route of the metabolism, we do see in both in vitro and in vivo experiments the same 4 DNA adducts formed that associate with the progression of liver tumors. And it's really the genotoxic carcinogenic endpoint that is the driver for the risk assessment of these PAs. And that is what the limit that Barbara highlighted, the 237 microgram per kilogram per day, is associated with. So these 4 DNA adducts are actually really important to remember, because I'll come back to them later as I reflect on the in vitro strategy that we've followed. Now, there are also N-oxide versions of the free base PAs. I'm not going to get too much into those today, although I think maybe at a subsequent BSC meeting, we might be able to talk more about N-oxides. They're very interesting compounds in respect of PAs. They represent both a detoxification of the free base PA, but themselves, they can be metabolized by the gut microflora to the free base PA. So there's a retroconversion that's pretty interesting that can be also activating for toxicity, but that's for another day. So this is just a high-level visualization of the strategy that we're following in P&G to really better understand this topic of potency differences in pyrrolizidine alkaloids. So we're really focusing on measuring the uptake in the gastrointestinal tract, looking at the biotransformation in the liver, and then really what's happening at the cellular level in respect of genotoxicity. And we're leveraging pharmacokinetic modeling, PBPK modeling, to really integrate the in vitro data that's coming from these different experiments to inform on relative potency. So I'm going to now step through a bit more of the in vitro data that we've generated thus far to parameterize this PBPK model, this physiologically based pharmacokinetic model. So firstly, GI tract permeability, as I mentioned, we're looking to measure that. Trying to understand the metabolism, both pre-systemically as well as in the liver. So we've looked to measure that. And then also the matter of intrinsic potency in the hepatocyte. So what is the fraction of reactive metabolites formed? And we've looked to measure that. So this is the data obtained from the CACO2 assay, which is a gut permeability assay. And we can see that different PAs vary in respect of their gut permeability. So these monoesters down here exhibit low gut permeability versus the diesters up here, which can permeate more considerably and fall into the moderate category of gut permeability. And this is actually what's also been reported in the literature. So we're seeing good agreement there. So the permeability rates not being equal means that we really need to consider this in our potency determination. The fraction that reaches the liver will differ according to which PA we're actually assessing. So the next thing we've looked to do is understand that question of pre-systemic metabolism versus liver metabolism. And these are experiments with microsomes, both intestinal microsomes and liver microsomes. And really the takeaway message here is that there's a negligible loss of our PA in the intestinal microsomes, and yet we see a substantial loss in the liver microsome. So that's really telling us that liver metabolism, is far more important than intestinal metabolism, and that becomes relevant when we parameterize the PBPK model. So the next thing we've sought to understand is this question of metabolic stability. At its simplest level, you might think, well, the rate of metabolism could influence toxic potency. But really, I think the takeaway message here is, please don't assume that. Metabolism alone is not an indicator of toxic potency. So what we've actually looking at here are, again, liver microsomes, rat liver microsomes in this case, across some different substrate concentrations. For the most part, we do see that these metabolic rates, as shown here, are concentration dependent, except perhaps for Ridley. But we don't see a correlation between this rate of metabolism relative to what we know about the potency of different PAs. So this little beastie here, lasiocarpine, is a fairly potent PA, whereas lycopsamine, the purple here at the end, is really not a very toxic PA. It's a monoester versus lasiocarpine, which is a diester. And yeah, the rate of metabolism would seemingly look fairly similar. So yes, metabolism matters, but it's not a driver for potency by itself. So what we then sought to actually quantify is the fraction of reactive metabolites. That is the driver for potency, this measure of intrinsic potency. So really the definitive data that we've looked to generate to input to the PBPK model is a measure of DHP DNA adducts. Remember, I said it's the same for adducts that correlate with liver tumours. We can measure those DNA adducts in vitro. We've measured them in rat sandwich culture hepatocytes relative to the AUC of the parent PA. So it's the ratio of DNA adducts over AUC that we're actually looking at on this graph. And you can start to see again that the different PAs we've measured really do vary in respect of their potency. Now, these experiments are analytically challenging. The synthesis of the standards needed for the DNA adduct quantification are challenging, though we're blessed here with some excellent analytical colleagues who certainly made this achievable. But we did actually look to see if there would be other ways to inform on intrinsic potency through more simpler measures. And so to that end, we actually explored log D as a surrogate for intrinsic potency. So log D reflects both degree of ionization and lipophilicity, which obviously relates to permeation and metabolism. And we pleasantly, I won't say surprised because it makes sense biologically, but we were pleased to see that the experimental values that were generated for log D correlated really nicely with our in vitro adduct over AUC measures. And moreover, that the predicted log D values using ACD or Perceptor software compared well with our experimental log D values. So this sort of tells us that where we don't have measured adduct over AUC data, we can use log D as a surrogate for intrinsic potency. Okay, so you're probably thinking, where's she going with all of this? Well, this is where we're going. We're talking about parameterizing a PBPK model to inform on the relative potency of PAs so that we can make more toxicologically relevant risk assessments of these PA mixtures as they occur in the exposures that Barbara highlighted. So in essence, and I won't be able to step through all the details here, again, we could get into a lot of detail, but in essence, we're looking to inform, on what the predicted liver AUC is, and to multiply that by our DNA adducts over AUC measures from the rat sandwich culture hepatocyte experiments. And that's what will give us a ranking of relative potency. So in essence, what are the predicted DNA adduct that would form from a given PA exposure by the time you've made this calculation? So we've started to build the PK model to inform us on, firstly, liver AUC for different PAs. We've done that in the first instance for rat, and we've done it with a few different PAs where in vivo data is existing. So we want to be able to verify our model predictions and understand if our model is performing well. And what we're looking at here is actually good model performance. So both the shape of these different curves relative to the measured data is within twofold, and that's a good place to be from a PBPK modeling perspective. So both the AUC and the Cmax predictions from the PK model that we've built using this in vitro data, is comparing well with in vivo data. So the next thing that we've started to do now that we have a model that's working for liver AUC predictions, is start to integrate with our DNA adducts over AUC data from the rat sandwich culture hepatocyte. We've done it for ritalin in the first instance because there we have measured liver DNA adduct data available to us. Again, looking to understand how the measured data compares with our predicted values. And in this instance, you can see that we're actually in really good agreement. So our model prediction is actually really very close to the measured data. So now we've got a model that we believe is predicting nicely the DHP DNA adduct formation. So the next step would be to look at taking this modelling approach across a series of PAs using our in vitro data that we've obtained. Remember, we've got the gut permeability data, we've got our liver metabolism data, and we've got our DNA adduct over AUC data, and we can start to integrate all of that into our rat PBPK model and inform on the differing potency across the PAs. The other thing that we want to do is explore the question of, well, what about low doses in humans? So our current PBPK model is built around rat, and that indeed makes sense because from a regulatory perspective, the limit that's set for PAs is derived from a rat bioassay. But we can use PBPK modelling to really explore the question of potency at low doses in humans. And we can do that to include the PAN oxides that I mentioned earlier, which arguably may be of lower potency than the free-base PAs. They certainly, from some of our experiments so far, would indicate they don't permeate the GI tract at a high level. But as I mentioned, they do undergo this retroconversion by gut microflora. So we'll be looking to extend our PBPK model to include an intestinal microbial compartment. Now, I just want to switch gears a little to reflect on some of the work that my colleague Stefan Fuller and his lab here at P&G have been undertaking. So this is a separate data stream. It's a endpoint that would be further downstream of our DNA adduct formation, particularly here we're talking about micronuclei formation as a measure of genotoxicity. So I wanted to highlight that, from the work that's been undertaken by Ashley Alamang in Stefan's lab, she's also shown that many different PAs vary in respect of their genotoxic potency. And this is using a human liver cell type, the HepRG cell, and a micronuclei formation. We're looking here at the confidence intervals of benchmark dose modeling that represents a twofold increase in that micronuclei formation. And you can see again that trend in genotoxic potency across different PAs. So it's not obvious to see from the slide here, but when we published this data, we did actually correlate with our rat sandwich culture hepatocyte DNA adduct data, and we see that there's a good correlation of the rank ordering of potency. So Stefan's actually working to continue to explore some of this question of genotoxic potency under low oxygen conditions, which becomes, again, particularly relevant for the N oxides. But again, I think that could be a story for another day. The other bit of the story I wanted to tell, however, today, with the genotoxicity studies conducted, is this question of what about the PAs in mixtures? So they are found as mixtures. They're not found as single PAs. There's generally more than one PA that has to be considered as part of that level, the threshold that we have to meet. So here we're looking again at the HepRG micronucleus assay and some mixture experiments of different PAs that span different structural configurations and have different potencies. And what Ashley's done is actually combine the PAs to achieve equal potency. So these are not equal concentrations, they're studies reflecting equal potency. And what we're seeing is that it is a matter of dose addition that is really sort of outputting. If we'd seen synergistic effects in these different mixture experiments, then these we've seen quite some significant deviation from this central line here. So we are seeing dose addition with the different PAs, which makes sense because they have common mode of action. Like I say, the 4 DNA adducts are consistently measured, and so it isn't surprising, but nonetheless it's reassuring to know it's dose addition that we need to consider. So yeah, just to wrap up really, I mean, PAs are pretty interesting botanical metabolites. They do undergo, reactive metabolism, and it's that fraction of reactive metabolites formed in the liver that drives toxicity, but as well we have to consider absorption in the gut. And we do see that not all PAs are created equal. I think I've shown you that from the in vitro data that we've obtained so far. And we really can measure those differences in vitro. So I think it's a really nice shout out to in vitro experiments that they can aid the assessment of these phytochemicals. PBK modelling is definitely showing promise to inform on the pharmacokinetics and the biomarkers of effects of different PAs. Again, so far we're seeing good agreements with in vivo data. That's important because we won't have in vivo data for all the PAs. So we use what we can, but the very reason we're taking an in vitro approach here is that there just simply isn't, and there will never be, the in vivo data for all the different PAs that Barbara highlighted. We're not going to have 28 different bioassays, so we really have to go in vitro here. But nonetheless, we use the in vivo data we have, and we are seeing good agreements with our modelling approaches, so that's reassuring. And like I say, the next step now is to start to actually inform on the potency ranking across different PA structures as a next step, now that we've got confidence in the model that's been developed. And then finally, just to remind that we've now tested and assured that the effects of these mixtures are additive, not synergistic. So that would really suggest that the risk assessment of PAs could and should be based on dose addition scaled for relative potency. Now, at this point, it's, I think, only right to come back to what Barbara concluded with. This work I've shared with you today really sits in a much broader context. There are academics and national agencies all working on the topic of PAs, and we've had 3 workshops now, and the most recent workshop is actually finalizing, while the manuscript has been submitted, actually a consensus paper from that workshop highlights 2 statements I wanted to highlight today. Firstly, it appears warranted to take into account IREP, so potency factors, in risk assessment of PAs. I think that's a really interesting place to be relative to where we first started with our workshop discussions. Reflects a lot of hard work by many people. And the other statement, it needs to be considered, however, that a consensus should be achieved among responsible regulatory bodies in order to better define the requirements and conditions for the regulatory use of such data. So said another way, scientific consensus is growing that not all PAs are created equal. We think we can derive potency factors that are scientifically credible. So now the question is, what can be done with that in a regulatory risk assessment setting? So final reflections. When we started 5 years ago, it felt like we had a mountain to climb. I think we've made it some way up the mountain with our in vitro testing approach and PBPK modeling, so considering exposure. And none of that would be possible without the in vitro measurements, the analytical experiments, the PBPK modeling. And I want to recognise my colleagues here that largely are pushing that peanut up that mountain. Thank you very much.
All right. Thank you so much, Catherine. Thank you for yours and Barbara's really complimentary talks. We have a couple minutes for questions. We have received one in the chat, so if folks can either put theirs in the chat or raise their hand, we can start going through. We had a sort of long question from Ibrahim. I'll divide it up into a few pieces. And Ibrahim, if I miss anything, you can come off mute. But he had a question about what about potentially new PAs or PAs we haven't found before? Is that on anyone's radar, or there's things we should look at in terms of like climate change?
Well, may I try to answer that? It deals about monitoring PA content in PA-containing plants, so not in contaminated material, but in plants which genuinely contain PA. Well, of course, that would be interesting to collect data to see if and how the PA content or the PA pattern might change due to it influences from environment, climate, and so on. To my knowledge, there is not such a database existing, but I think that would be of high interest. With regard to our database, it contains only data on contamination, except very few data on homeopathic mother tinctures of PA-containing plants, which are used in homeopathy in very high dilutions. But we do, from our point of view, there is no data existing about that. But maybe somewhere in pharmacognostic research or whatever, but I don't know a database.
Thanks, Barbara. That's helpful.
Mm.
Catherine, I don't know if you have, if you can see the question, anything to add either on new PAs or databases of that identify the source of a PA, like databases on the plants?
Yeah, I'm not aware of a database. PAs are fairly well understood, and there are many, as Barbara highlighted. There's also, I think, some work being done looking at plant DNA, and I guess one could, in relation to PA, one could sort of track that a bit and see what was possibly shifting over time as a consequence of environmental factors. But I guess, PAs have sort of evolved to protect plants from predators, right? And environmental factors have been present in plant growing for many centuries. And so, I really don't know how much it would likely vary as a consequence of what is yet to come. It's an interesting question for sure.
Great, thanks, Catherine. Any other questions or comments for our keynote speakers? I really liked in your talk, Catherine, the use of both the PBPK modeling and the in vitro talks. Have you all at P&G maybe looked into different results from different PBPK modeling and thoughts about how that might, the different compartmental models might change the results you get?
So, yeah, I mean, firstly, I think it's probably only right to point out we're not the only ones working with PBPK modelling for PAs, right? Ron Beecham's group at Wageningen University is also working in this space, and we're discussing with Yvonne different PBPK modelling approaches to really understand that very question. I think when we get into the N oxides, it certainly will possibly matter what compartments you have. Like I say, the intestinal microflora is a pretty key one for the N oxides, and its intestinal metabolism is less relevant. It's really quite negligible for free-base PAs. So yeah, one has to step through what matters most in respect of absorption, metabolism, distribution, excretion in order to define what experiments, what compartments you actually need to think about in the model predictions.
No, that's really helpful. And I liked in your talk, you emphasized, we have animal data for some, but we won't for all. But so can we leverage those learnings for the new, the understudied ones?
Definitely.
And then Corrado just had a clarifying question. He put in a statement. I missed whether there was a correlation between the DNA adducts in the liver and the genotoxicity. Oh, but it looks like Stefan just answered too. But go ahead, Catherine.
Yeah, no, definitely there is. I think I mentioned I wasn't showing the direct comparison in the slide, but we talked about it in the publication, and there is a good correlation in the rank ordering of potency using the DNA adducts data as well, genotoxicity data. So yes, there is a correlation, a strong correlation.
Great, thanks
and
for
we
that.
probably need a better figure, Stefan, to show that.